Solid tumors are surrounded by benign tissues that influence and are influenced by the tumors. These surrounding benign cells, which include vasculature, immune cells, neurons, and other cell types, are collectively known as the tumor microenvironment. Tumors manipulate their microenvironment for the benefit of the tumor. For example, it has been well documented that autonomic neurons innervate and drive malignant growth in a variety of solid tumors. However, the mechanisms that underlie these neuron-tumor relationships are not well understood. Our commentary discusses recent work by Amit et al. That describes trophic relationships between oral cavity squamous cell carcinomas and nearby autonomic neurons, and how these relationships arise through direct signaling between tumors and local neurons. In this work, 4-NQO was introduced into the drinking water of p53 knockout mice to induce growth of oral cavity squamous cell carcinomas. These carcinomas were then used as a model for tumor microenvironment interactions. Using this model, this group discovered that loss of p53 expression in oral cavity squamous cell carcinomas resulted in increased nerve density within these tumors. This neuritogenesis was controlled by tumor-derived microRNA-laden extracellular vesicles. Specifically, microRNA 34A delivered by extracellular vesicles decreased neuritogenesis, whereas microRNAs 21 and 324 increased neuritogenesis. The neurons innervating p53 deficient oral cavity squamous cell carcinomas were predominantly adrenergic and arose through the transdifferentiation of trigeminal sensory neurofibers to adrenergic neurofibers. This transdifferentiation corresponded with increased expression of neuron reprogramming transcription factors, including PAL5F1, KLF4, and ASCL1, which were overexpressed in the p53 deficient samples and are also proposed targets of microRNA 34A mediated regulation. Finally, human oral cavity squamous cell carcinoma samples enriched in adrenergic neuron markers were found to be associated strongly with poor outcomes in patients. This commentary highlights several important findings for the field of extracellular vesicle-based signaling. First, these findings demonstrate that tumors and local neurons within the tumor microenvironment are dynamically changing and responding to each other. Secondly, these data illustrate that microRNAs delivered by extracellular vesicles control these dynamic relationships formed between neurons and tumors. Finally, we highlight that this novel relationship between tumors and neurons will be important for developing new models and new therapies to treat patients who have cancer.